Okay, AP Bio, let's wrap up this chapter where we looked at first X-linked traits, and then we looked at linked genes and how to calculate uh, cytogenic maps. Thank you for the English. Now, as this really gets us rolling on the chromosomes determined to be those heritable factors, those the inheritance that Mendel called heritable factors. Let's start as we're going to look at next the structure of DNA, RNA, how it copies itself, how proteins are made, et cetera, et cetera. Let's also note that, we saw crossing over. We've seen independent assortment crossing over random um, mating stuff leading to variation. But we should also talk about crossing over other stuff and alterations in number or gene number or chromosome number that can cause disorders besides X-linked, besides stuff, besides whatever. Now, huge chromosome alterations in humans and mammals often does not lead to a viable offspring. Often those are miscarried or cause a variety of developmental disorders that don't allow that offspring to survive. Plants, on the other hand, have evolved and developed to a way and adapted to a way that they tolerate genetic changes better than animals do. Um, and you can look in, well, there's reasons for it that we're not going to discuss here, but they're just able to tolerate, things like polyploidy. It would not be uncommon for plants to have a whole extra uh, triploid set instead of being diploid, but we'll talk about that in a minute. One of the big ones, if we're coming off of our mitosis meiosis discussion, is non disjunction, where the pairs of homologs do not separate normally during meiosis in meiosis 1. So as a result, one gamete will get two of the same type of chromosome, and another gamete gets none. And so you see the example here in meiosis 1 of a non disjunction. For some reason, the microtubule did not attach to the kinetochore. And so when they went through meiosis 1 and separated, those homologs didn't separate. In fact, they didn't separate until um, meiosis 2 with the non disjunction. And so you see what happens is, um, you know, you'll have a couple of gametes with um, trisomy. If the if it occurs in meiosis 1, you'll have two gametes with trisomy and two of them that are monosome. If it happens in non-disjunction happens in meiosis 2, then you'll have one gamete with trisomy and one that's monosomic and two that are standard. And trisomy is the term three genes. Aneuploidy results from the fertilization in gametes then in which non-disjunction has occurred because the offspring of that condition will end up with an abnormal number where you're supposed to have one from mom and one from dad. Now you have either two from mom or two from dad besides the one from mom or dad. So monosomic zygote has only one copy of a particular chromosome. Trisomic zygote has three copies. And here's where we talk about Down syndrome, because as you look in that karyotype down at 21 here, you have three chromosomes for uh, number 21. Down syndrome, one out of every 700 births in the US. And that changes to one out of 350 after age 35, 1 to 100 at age 40, and 1 in 30 at age 45. So we said a normal, and I think those numbers have gone a little bit 1 to 700 or 1,000, but you can see that you hit age of 40 and you in a sense dropped an exponential drop there. You dropped by quite a bit to a 1 to 100 or 1 to 30 deal. So why does that frequency go up? The belief is that and with the maternal age, with mom aging, and at 45, you're getting much closer to um, menopause. That some of those checkpoints, like the anaphase, the anaphase, where the kinetic core should be attached. Boy, I can't speak today. I'm sorry. Um, is not functional, and it blows through that checkpoint, and we still go to anaphase instead of being held at metaphase. Polyploid, as I said, is a condition in which it's not just one chromosome, but the whole complete set. So being triploid, having a whole extras as opposed to diploid, having a third set, or a tetraploid, having four sets. Very common in plants, not really in animals. And the polyploids are more normal in appearance than the aneuploids because it's better to have all of them than just one of them. Now, again, this is not necessarily true in humans because it's rare in humans, but plants, it actually leads to a greater variety of plants. Um, and they're able to tolerate it. Okay? Non-disjunction of sex chromosomes produces some other aneuploid conditions. This slide's a little out of order, but you can see there Klinefelter and Turner syndrome based on being trisomic or monosomic. Okay. Now, the last thing is of course DNA is being assaulted. I should maybe read out of Billy Bryson and stuff that talks about what a brutal uh, environment the inside of the cell is, and especially for DNA that's getting broken and repaired all the time. So, and we will see plenty of this in the future when we look at shifting reading frames and what mutations do. But just know that you can have chromosomal breaks in structure and they try to get repaired, but sometimes you have a deletion and a whole chromosome segment is gone. Sometimes you have duplication and you repeat that segment. Sometimes you invert it, it breaks and it comes reverses back in orientation. And sometimes it translocates where it moves to another chromosome. So there's an example of deletion. Chromosomal segment is gone. That's obviously going to alter things in gene expression. You can see duplication where you copied the BC, duplicated it. There's your inversion where it flipped around. And there's your translocation where all of a sudden you throw off the chromosomes themselves from being specifically. Anyhow, you see what you're saying. It moves from one to a non-homologous chromosome. And then, really, last, we'll talk about epigenetics. Epigenetics are things that are not changes in the DNA sequence. And we'll see more epigenetics in the future, kind of heritable traits that aren't DNA based. One of these we talk about right here is genomic imprinting because we're in that section where we talk about chromosomal inheritance. For some, the phenotype depends on which parents pass along the alleles for those traits. Because in gamete formation, they're imprinted or stamped. Such variation is called genomic imprinting. It happens in autosomal cells during gamete formation, and they're stamped with an imprint that says you're the one that is going to be, or you're going to be off, versus on. Okay. So here's a growth factor. Normal IGF allele is expressed from the father. The mother's normal chromosome is not expressed. And you get a normal wild type mouse. Okay. Now, if you get mutant IGF allele inherited from mom, and the normal IGF is expressed and the mutant is not expressed, then you will get a normal sized mouse. But if the mutant is expressed and imprinted from the father, then the normal one is not expressed, then you get a dwarf mouse. It appears that imprinting, and this is what segues us into our next discussion of DNA, It appears that imprinting is a result of methylation. Methyl groups will condense chromosomes. Now, if you think about it, as we talked about, when cells specialize, they turn off portions of the DNA sequence, and that's what we're kind of heading into. One of the ways to do that is methylation. Anyhow, so that's one way to methylate. You cysteine nucleotides get methylated. That essentially turns them off. Thought to affect only a small fraction of mammal genes. Most are critical for development. And then last couple things, we'll talk about extranuclear genes or cytoplasmic genes. They're found in organelles in the cytoplasm. Mitochondria, chloroplasts, other plant plastids. Plants have what are called plastids, carry small circular DNA molecules, and they are inherited by the mother through the zygote because, again, the sperm only brings the genetics and doesn't contribute any cell stuff. It always comes from the ovum. First evidence came from, essentially, call it calico plants. It's not calico plants, but just patches. There we go. Variegated plants. And we've seen when we looked at resistance and stuff with bacteria that they carry plastic, they carry plasmids and plastids, and we'll see more of those in the future. Okay. And then, of course, because of our mitochondria, as we talked about with pedigrees, some defects in mitochondrial genes prevent the cells from making enough ATP and results in diseases that affect the muscular and nervous system. Mitochondrial myopathy, optic neuropathy, things that affect the ATP. Okay? So there we go with things other than linked genes or X-linked genes and any of the disorders they may have. That's just in the chromosomal inheritance, things that break down and change. All right. Have a great day. We will talk to you later.